sorry. I just get emotional about proteins. I just love them so much. I love protein so much. But we're gonna continue our discussion in this episode on levels of protein structure. So, in order for a protein to function, we touched on this a little bit earlier, but in order for a protein to function, it must bind to a specific ligand. So, ligand is anything that binds to a protein, but in order to function, it must bind to a specific ligand. So, there's four basic levels of protein structure, and we're gonna run through them. They're primary structure, secondary structure, tertiary structure, and quaternary structure. And this is tough for me to do sometimes. But the pinky wants to stand up, so we're gonna go like this: primary structure, secondary structure, tertiary structure. That's kind of tough too, and quaternary structure there. So four levels of protein structure. The first one we're gonna discuss is the primary structure. So it's as we know, proteins are a bunch of amino acids bound together via these peptide bonds. So between each amino acid is a peptide bond, but a primary structure in the levels of protein structure is a linear sequence of specific amino acids joined together by these peptide bonds. So it's just one chain of amino acids, but if you change either the specific arrangement or different types of amino acid, it creates a whole different primary structure there. So remember those R groups are the major differentiation factors between amino acids, but taking a step up that ladder, if you change amino acid A with a different amino acid, say C, it's going to have a whole different primary structure. Or if you change the sequence, so let's say it's A, C, D, B, there's obviously actual names of amino acids, but if you move the D first with the A, it's going to have a whole different primary structure and be a completely different protein there. But in the primary structure, proteins are not functional, so they aren't able to function properly, whether as an antibody, whether as a clotting agent, whether as an enzyme. They just can't function whenever they're in this primary structure. But that's the least complex structure, or least complex level of protein structure. But then a little more complex than the primary structure is a secondary structure. So secondary structure is when there's a hydrogen bond between an alpha carboxyl and an alpha amine of non-adjacent amino acids. So non-adjacent, they're not right next to each other. It's when the amino acid or the alpha amine of an amino acid over here binds with the alpha carboxyl amino acid over here via hydrogen bonds, and it creates a kind of a helix shape, so like a spiral staircase or a helix or an accordion, one might think of, or the accordion. I like to think of the helix just because it makes a little bit more sense to me of this amino acid combining to this and kind of making a nice twirled little candy cane structure there. But once again, although secondary level of structure of a protein is more complex than primary, it is still not functional. So even more complex than a secondary structure is a tertiary structure, which is when there's a hydrogen bond between R groups of non-adjacent amino acids. So R groups, we discussed those in the amino structure of an amino acid episode, so check back on that. But a tertiary structure is when there's hydrogen bond between non-adjacent R groups. So R groups of two different amino acids that are not next to each other bind together, and these are definitely more complex than primary and secondary, and sometimes in tertiary structures proteins are functional, sometimes not, but sometimes proteins are functional when they're in their tertiary structure. Now, last but certainly not least and most complex is the quaternary structure, the quaternary level of a protein structure. So quaternary level is the most complex, and it's when there's when two or more polypeptide chains that are in their tertiary structures bind together. So it's essentially when tertiary structure, so there's we just discussed that when there's a hydrogen bond between R groups that are non-adjacent amino acids, between non-adjacent amino acids. Tertiary structure, in another tertiary structure, when two or more of those, or maybe three tertiary structures bind together, it forms this very, very complex shape, and it's the quaternary structure or the quaternary level of protein structure. So it's even more complex than tertiary because there's not one tertiary, but maybe two, maybe three, maybe four, all bound together into this extremely complex, shape. And in the quaternary structure, proteins are always functional. So proteins, just run back through. There's a primary structure, which is not functional. It's a linear sequence of amino acids. There's a secondary structure, also not functional, a little more complex than primary, but it's when there's hydrogen bond between non-adjacent alpha amine and alpha carboxyls of non-adjacent amino acids. Then there's the tertiary structure, which is when there's hydrogen bonds between the R groups of non-adjacent amino acids. And then last but certainly not least, the most complex, which are always functional. Tertiary are somewhat functional. They're sometimes functional proteins, but quaternary are always functional proteins. It's when two or more polypeptide chains that are in their tertiary structures bind together. So we got a tertiary structure, maybe even a third or fourth, all bind together to form this quaternary structure. It's an extremely complex shape, and it makes the protein functional there. So the ligand binds to the specific ligand binds to the protein and allows it to function accordingly there. So seems complex at first, but once you kind of understand that the primary is least complex, secondary is a little bit more complex, tertiary is even more complex, and quaternary is the most complex structure of protein. The more complex the shape, the structure, the more likely it is to be functional. But tertiary structures sometimes are functional. Quaternary, when proteins are in their quaternary structure, always functional. If you have any questions, type them in the comments below, and we'll get it all sorted out. But no analogies in that episode, but it's just pretty much spitting information to get up in the old brain there. So repetition is key. So work with these concepts. I guarantee they'll make sense here.